two? Is there supposed to be a third? Yeah. Okay. Good morning. My name is Nipun Merchant. I'm here from the University of Miami. Sorry for the weather. We tried to get you better weather, but yesterday was not too bad, at least. It's a pleasure to be here with my co-moderator, Dr. Su. And we have a really great session with some amazing speakers today, talking about the tumor microenvironment and drug resistance in pancreatic cancer. So we'll get started with our first speaker. Our first speaker is Manuel Hidalgo. Dr. Hidalgo is currently the chief of the Division of Hematology and Oncology at Weill Cornell Medical Center. He's also the Hugh Luckley Distinguished Professor of Medicine there. Dr. Hidalgo received his medical training and PhD training, as well as medical oncology training in Madrid. He then came to the United States, where he did a fellowship at the Institute of Drug Development in San Antonio, where he stayed on as a faculty member initially. Since then, he's moved to Johns Hopkins as faculty and BI Deaconess until his current role at Weill Cornell. As you all are aware, his main focus of research has been in new drug development in pancreatic cancer, and his group has also popularized the avatar mouse model in cancer, which you're all familiar with. His current work focuses on strategies for personalized medicine and immunotherapy in pancreatic cancer, which is what he'll be talking to us about today.
Good morning. Thank you very much for the invitation to give this talk on pancreas cancer immunotherapy. These are my disclosures. And today what I would like to do is cover the current status of immunotherapy for pancreas cancer, then discuss some of the proposed mechanisms of immune resistance, and finish with some new therapeutic interventions in this field. Cancer is treated these days by these 5 main modalities: surgery, radiation therapy, chemotherapy, precision therapy, and immunotherapy. And while precision therapy and immunotherapy have made significant impact in the management of different tumor types, such as melanoma or non-small cell lung cancer, pancreas cancer remains resistant to these strategies. Very few patients are candidate for targeted agents, and we still treat them with surgery, radiation therapy, and chemotherapy with not very good results. Immunotherapy, in addition, has basically no activity in this disease, as we know. There have been multiple studies testing the role of checkpoint inhibitors alone or in combination, different checkpoint inhibitors combined or combined with chemotherapy in pancreatic cancer. I won't spend time summarizing these studies, but the conclusion is that these agents, as we know them today, PD-1, PD-L1, CTLA-4, are basically ineffective. As an example, this is a randomized trial testing durvalumab plus tremelimumab versus durvalumab in the patients with the advanced pancreatic cancer previously treated with chemotherapy. And as you can see, there is no evidence of benefit combining the 2 agents versus single agent. And in addition, and more importantly, the results are very, very poor and very limited. For this reason, none of these strategies has gained any kind of regulatory approval in this disease. Now, the mechanisms by which pancreas cancer is resistant to immunotherapy are diverse. Every day we see papers, new papers that are proposing new mechanisms. Some of those, like the IDO pathway, IL-10, have been already tested in clinical trials and have not shown to be effective. Listed in this slide, there are some others that I will discuss, and I will discuss some of them today. But there is significant interest in bringing new drugs into this disease. And as you can see in this review that we wrote a couple of years ago, many of the new agents that we're trying to explore are immune-mediated strategies and work by an immunotherapy based kind of mechanism. Multiple studies with many of these drugs. I have selected a couple of them that we have new data with, including CXCR4 inhibitors, some other stroma targeting strategies, viral therapies, and I will finish discussing new approaches for cell therapy. One of the fundamental mechanisms by which pancreas cancer is thought to be immune resistant is related to the tumor microenvironment and the pancreas cancer stroma. There is data that shows that cellular elements in the stroma, mainly the cancer-associated fibroblasts, produced cytokines and substances that are involved in the immune suppression. One of those mechanisms is the CXCR4, CXCL12 pathway. The proposed mechanism based on preclinical studies is that cancer-associated FAP positive fibroblasts secrete cytokines CXCL12 that binds and activates CXCR4, and by that mechanism prevents T cell entry into the tumor. So the tumor does not respond to immunotherapy for the simple reason that there are no T cells to be activated in the tumor microenvironment in close proximity to the malignant epithelium. In preclinical studies conducted by Doug Fearon and his group, he showed that by blocking different elements in this pathway, you can get these cells into the tumor, and that is sufficient to then make the tumor sensitive to PD-1 blockade. We have explored this strategy in the clinic using a peptide that blocks CXCR4. This is the BL8040 multisaffordide. And we showed in paired tumor biopsies in patients with advanced chemotherapy-resistant pancreas cancer that blocking this pathway is indeed sufficient to get T cells into the tumor, as you can see here in these paired tumor biopsies from pre-treatment and after treatment. This confirms and supports the preclinical data observed in mice, and this strategy was advanced to clinical development in combination with Pembrolizumab and chemotherapy in the second-line setting. These were patients that had received one prior line of chemotherapy. And as you can see, there is some evidence of activity shown by responses and stable diseases that, as we observe often with chemotherapy, they tend to be durable. This resulted in a median overall survival of 6.6 months and a median progression-free survival of 3.8 months. That while reasonable is not clearly better than what we see with chemotherapy alone. So for that reason, I think this regimen is sort of a step forward, but it needs additional fine-tuning and additional evidence of activity before we can take it to next phase 3 type of registration trials. We have continued to be interested in adding elements or inhibitors to this pathway, and we are currently working in preclinical models with this agent, which is quite interesting. This is an FAP targeting antibody drug conjugate, and we have shown in some of the preclinical studies in pancreas cancer xenograft that is very, very effective, and as expected by an ADC, is effective as a single agent, which is kind of important. And the mechanism appears to be that the drug binds to the FAP positive fibroblast, then releases the payload tubulicin, and that tubulicin is effective in one end eliminating fibroblasts, and for that reason you get additional T cell infiltrate, but also there is a bystander effect and you're able to eliminate malignant cells. So this drug is about to enter phase 1 development. Most of the data is published in this paper in Clinical Cancer Research. I want to spend more time in discussing it, but it's moving forward as a single agent in combination with chemotherapy, and importantly, and you can read that in the paper, has very nice synergistic effects in combined with Pembrolizumab. So we will sort of reinvent or redo sort of the combat trial strategy now using this new approach. In the same kind of mechanistic approach, we have also worked with an agent that it is very, very interesting as well. This is a adenovirus that has been engineered for 3 important things. 1, selective replication in tumors. B, liver detargeting. As you know, many adenoviruses, when administered systemically, land in the liver, they replicate there, cause hepatitis, inflammation, but really don't reach the tumor. So this one is supposed not to, or designed not to do that. And then what is very interesting, though these days also conflicting, is that it produces hyaluronidase, and by that mechanism decrease tumor stroma content of hyaluronic acid, which has been proposed as one of the barriers to chemotherapy and immunotherapy delivery. Now, we know that the studies conducted with pegylated hyaluronidase, while there was supporting the data from the phase 2 studies, didn't materialize in the phase 3 study. So whether or not how relevant this target is at the moment, as I said, there is conflicting information, but this virus, it replicates when administered sub-Q in mice. The replication is maintained for up to 22 days, has intrinsic anti-tumor efficacy, degrades very nicely hyaluronic acid, and by that mechanism facilitates chemotherapy and immunotherapy delivery, and has been effective in broad range of preclinical models. This paper is about to be published, will be published in the next week or 10 days, is already accepted for publication. This was taken to the clinic in a study in which the virus was administered IV, which is very important. And you can see here that day 8 after treatment, it lands into the tumor, and you can detect it there, and it replicates in the tumor tissues. And this study has been already, as I said, conducted, about to be published. We've seen some patients with very nice evidence of anti-tumor responses, combining the virus with chemotherapy, and it's being shown that there is immune infiltrate and immune-mediated mechanisms in the tumor, in paired tumor biopsies, and the overall median survival of this study has been 13.5 months. And again, we're wrapping this up and getting it ready for additional registration trials. Just to finish, I want to give you a couple of slides or a couple of data on cell therapy, and an approach that we've been developing in the lab, also about to be published. It's coming out in the next few days, in which we have used organoids taken from patients with pancreas cancer, and developed in collaboration with Senthil Muthuswamy at Harvard Medical School. We have combined in an autologous system with peripheral blood mononuclear cells obtained from the same patient. And then we use this co-culture to generate cells that we call OPT cells, organoid prime T cells. And the question is, do these cells that have been now free of any immunosuppressive signal combine with the tumor and cytokines, will these cells become sort of effective in tumor, recognize the tumor, become effective in tumor killing? It's sort of an approach to develop ex vivo or TILs, if you like. And what we have seen is that, yes, plain PBMCs that have been taken from the patient, but have not been co-cultured with the organoid are not effective in recognizing the organoid and eliciting an immune response. But when you do the OPT cells, these T cells, 15 days after the co-culture, they proliferate, they secrete interferon and granzyme B. And when we take these OPT cells made with an organoid and treat an organoid from the same patient, another organoid from the same tumor, 20 hours later, there is a massive cell killing. So they get activated, they are immune reactive, and they kill cells. And what has been also observed is that while, as expected, non-co-cultured peripheral blood mononuclear cells from a given patient, they contain a very large repertoire of T-cell receptors. After the co-culture in these conditions, there is a very nice oligoclonal selection. And in this particular patient, 5 clones represented up to 99% of the TCRs. And we have then taken an organoid depleted and an organoid enriched TCR engineered SKW3 cells. And as you can see here in the autologous system, when we then put these cells in front of this patient tumor, the organoid selected TCR activates the immune system, the depleted does not, and in the allogeneic system, it does not, suggesting that there is a specificity and that these TCRs are recognizing the tumor. So we're using this platform now in 2 strategies. One is to take them to the clinic as OPT cells, period. We're working on making the organoid process sort of regulatory compliance and GMP compliance and to shorten the time for production of the cells. But this is like using TILs, that there are already some studies, very nice studies published in melanoma and non-small cell lung cancer. The only thing is that instead of taking the lymphocytes from a tumor biopsy and expanding them, we are sort of making them ex vivo by co-culturing with the organoid. And one thing that is in the paper, and you can read when published, is that and I said in about 2 weeks, is that these cells have a non-exhausted memory phenotype, which is also important because may indicate that they will be sort of persistent for long. And of course, this is also a platform to just plainly discover tumor effective TCRs, and we're working on that as well to determine whether or not these TCRs then can be used to engineer T cells and they will have anti-tumor activity. Just to finalize, how are we moving all these sort of complementary but different treatments into the clinic? One strategy is this trial called Precision Promise, which is designed this is a PanCan sponsor multi-center clinical trial. Many institutions in the country are participating, and it's an adaptive randomized design by which we take 2 control arms for FOLFIRINOX and gemcitabine nab-paclitaxel, and again add investigational phase 2-3 arms in a 2-stage design by which we combine standard of care chemotherapy with new agents. Many of the ones that I mentioned to you before will enter this pipeline, and the randomized adaptive design is kind of nice because you can advance those agents that work, but the system sort of eliminates those that have less activity. So I think this is a very interesting approach to test in a more rapid and efficient manner agents that preclinically look promising, early clinical studies look promising, but then we have learned over and over again that when we take them to sort of randomized clinical trials, they just don't work, and it's important to learn that in an efficient and rapid manner, and hopefully, of course, some of them will work and will change the standard of care. So to conclude, CPIs are not effective in PDAC, that's known. There have been multiple mechanisms that are involved in immune resistance. Every day we learn about a new one, and And it's important, and I think more work needs to be done to define which ones are really the key ones. And there's a plethora of agents and strategies in development. So we need priority and rapid effective evaluation. And I think the precision promise approach is one sort of the ways to get this done. I want to finish by acknowledging the different team members that participated in this work, and thank you very much for your attention.
I think we'll hold off on questions till the end of the session, and we'll go to the next speaker. So the next speaker is Dr. Marina Pasca Di Magliano, who received her undergraduate degree from Naples, Italy. And following her graduation, she did an international PhD program in Austria, Vienna. After which she moved to the United States to work in Matthias Hebrock's lab, where she studied pancreas development and also started looking at hedgehog and Wnt signaling in pancreatic cancer. She was then recruited to the University of Michigan to start her own laboratory, where she has made significant contributions to the field, specifically in relation to developing a inducible and reversible iKRAS mouse model in pancreas cancer. And her interests have long focused on cellular crosstalk between tumor cells and the tumor microenvironment, with a particular interest in the immune regulation of pancreas cancer, which is going to be the focus of her talk today. Thanks.
First of all, I would like to thank the organizers for giving me the opportunity to present my lab's work. And I've greatly enjoyed being back at an in-person conference. So my lab, just like Nipun Laisi said, is interested in how the different cells within the microenvironment talk to each other and how that regulates tumor progression and as well as resistance to treatment. And part of why we are interested in that, I have to move a little because I'm short and I can't see the slide. Part of why we're interested in that is what many speakers before me have referred to, the complexity of the microenvironment in pancreatic cancer. You can see here on the left a slide of a normal human pancreas populated mostly by acini, and about in the middle you can see a normal duct with the fibroblasts that surround it. But to the right side of the slide, you can see how in a tumor, the majority of the volume is occupied by this very complex microenvironment. And what I wanted to tell you today about is newer work in my lab, where we decided to map the microenvironment of pancreatic cancer using human samples rather than mouse. But before I get to that, this slide is just a big picture overview of the different questions my lab is interested in. So we want to know what are the components of the microenvironment, how they are regulated by tumor cells, and how in turn they regulate carcinogenesis. Okay, so this is a scheme showing how we went about conducting this work, and we started in fall 2018, and that turned out to be lucky timing because we were able to switch part of my lab to working with human samples and doing a lot of bioinformatics. And as you can all imagine, that turned out to be great when we had a lab shut down and we were able to keep working. So to map the microenvironment in human pancreatic cancer, we use a multimodal approach. On one hand, we dissociated the tissue and looked at gene expression by single-cell RNA sequencing. We also looked at protein expression by mass cytometry. And then we also, whenever possible, use slides to stain the tissue and validate our dissociated tissue findings on the actual original tissue that had not been dissociated. And that is particularly important because the microenvironment is heterogeneous in different areas of a tumor. This was a team effort, and you can see here the people in my lab that did the work. Those are Nina Steele, Eileen Carpenter, Sam Kemp, and Viren. They are, Nina is a postdoc. She's about to start her own lab. in January. Eileen is now a faculty member who stayed at Michigan. Sam is now a postdoc. She was a graduate student at the time. And Viren is about to graduate soon. And we had, of course, bioinformatics support by Arvind Rao and his group, and a number of faculty collaborators, including basic scientists and clinician scientists. So at the time, all the data that was available for pancreatic cancer sequencing had come from resected tumors. And we decided that we wanted to be sure to look at tumors of patients that were not resectable. And therefore, we use a combination of fine needle biopsy samples, as well as samples from Whipple or from distant pancreatectomy. And then whenever possible, we acquire adjacent normal tissue. This slide is what went in our original publication. We now have quite a few more samples than that. We started with mass cytometry, which is a version of flow cytometry, which allows to, instead of using fluorophores to label the cells, we use heavy metal isotopes. And the advantage of that is that we can use a lot more antibodies and therefore map the cells much more in detail. You also need much fewer cells compared to normal flow. And as you can imagine, since a lot of our samples came from biopsy, we didn't have so many cells to start with. And I'm not going to get into the site of data very much in detail, but I just wanted to give you a big picture. You can see to the left, those are all benign pancreas samples. And you can see that by and large, they're populated by those purple cells. And because of the way our panel is designed, that purple color reflects both tumor cells and fibroblasts. While the pancreatic cancer samples to the right have a variable but extensive immune infiltration. And you can also appreciate that there is quite a bit of variability from patient to patient. To define the cell populations that were in each tumor, we use single cell RNA sequencing, and you've seen in a few maps, so I don't really have to explain this, but generally each color, so the way the cells are projected is unbiased, and then we can go in and look at marker expression so that we can attribute each cluster to a cell type. And you can see that we find, of course, tumor cells and a large number of infiltrating immune cells, which was the focus of this study. And also, we also detected fibroblasts. The way we processed the tissue was optimized to get as many immune cells as we could. And unfortunately, you can't really both optimize for immune cells and fibroblasts. So in our case, we are skewing our tissue composition to immune cells. I'm going to specifically hone in on the CD8 T cells, which, as you all know, are the cell type responsible for anti-tumor immunity. So when we compared we took out the CD8 T cells, and you can see to the left the distribution of effector and exhausted CD8 T cells in no malignant tissue. And maybe I should preface this by saying that the no malignant in this case was surgical samples that were in one case adjacent to pancreatic adenocarcinoma, and in two other cases adjacent to other neoplastic conditions that require part of the pancreas to be excised. And you can see that there is, if you compare the adjacent normal to the pancreatic cancer samples, there is a shift in the composition of CD8 T cells, whereby in the adjacent normal, you have more effector T cells, but in the tumor, you have an enrichment of the exhausted T cell populations. And while those populations came from unbiased clustering, and then they express a large number of markers of both exhaustion and activation, I'm only showing you two of those markers, granzyme B and EOMs, to show you how the cells are different. We then map that across all our samples, and you can see that if you look on a patient-by-patient basis, it is pretty consistent that there are relatively few effector T cells in the tumor compared to the adjacent normal area, while there is a very large increase in exhausted T cells. So we were particularly interested at the time in understanding the composition of immune checkpoints since, as you've nicely heard in Dr. Hidalgo's talk, some immune checkpoints have been tried in pancreatic cancer and they were not effective. And so we mapped not all immune checkpoints, but a larger subsets of immune checkpoints that had been well characterized on the T cells. And then we looked at their ligands on non-immune cells. And what we found was that, of course, we saw expression of PD-1 and other immune checkpoints, but we observed that TGF was very highly expressed in pancreatic cancer CD8 T cells. And not only was highly expressed, but it was the only one of the checkpoints that really identified the exhausted T cells. So we then use an algorithm to predict potential interactions between different cell types in the microenvironment. And the way this works is that the algorithm is looking for expression of ligands and expression of receptors in different cell types. And so if a type of cell has a receptor and the other one has the ligand, it will predict that they potentially interact. Of course, it doesn't tell us that the interaction is occurring, but it gives us a hypothesis we can then go and validate on the tissue. So in that case, we're plotting some of those predicted interactions. And it is a little bit hard to see, so I use those big arrows to highlight that among those, the interaction between TIGIT on the CD8 T cells and PVR actually on several other cell types. This is showing macrophages, but PVR is also expressed by tumor cells themselves, endothelial cells in the microenvironment. And it is shown as an interaction that's upregulated in the malignant samples versus the non-malignant. That's of course just RNA, and so the first step was to verify whether TGF was expressed at the protein level. And so for that, we went back to our mass cytometry data where we luckily had a TGF antibody. First of all, you can see to the left that just like I showed you in sequencing, PD-1 and LAG3, two other immune checkpoints, were not enriched in T cells in the malignant samples versus controls. But on the other hand, TIGIT was enriched in the CD8 T cells of almost every patient that was included in our cohort. And the graph to the right just shows how the data looks. We also, and I don't have it here, validated expression by immunostaining, and we could see TIGIT expressing T cells adjacent to PVR expressing non-immune cells or non-T cells. Then we thought we wanted to see whether the increase in TIGIT in T cells in the tumor correlated with an increase in the T cells in the peripheral blood of the same patients. And so for all of our samples, we had also collected blood and we had sequenced PBMCs. And TIGIT was actually the only immune checkpoint where we found a good correlation between tissue and PBMC levels, which is potentially interesting because a patient with high TIGIT in T cells in the blood might indicate a patient with high TIGIT in the T cells in the tumor. And I'm sure many of you have seen a very recent paper from Tyler Jack's laboratory where they actually did some functional validation of TIGIT and showed that it is a driver of immune suppression in pancreatic cancer. So it was nice to see that. So this is, the conclusions from that first part of the study, but I don't have a timer. Do I have a few more minutes? So I just wanted to show you some follow-up work where we went into one of the prevalent cell populations in pancreatic cancer, which is the very large population of myeloid cells. And those myeloid cells are a mix of macrophages and cells that are called that are granulocytes, but they're also referred to as myeloid-derived suppressor cells. There is a little or immature myeloid cells. So those are labeled here as granulocytes. And you can see that there are heterogeneous populations of those cells. So again, we wanted to know how those myeloid cells in the tumor are different from the myeloid cells in benign tissue. And we we took out just the macrophage object, decided to focus on that, and we plotted. And again, by unbiased population defining, we found two main populations that we called, for reasons that I won't get into, they are referred to here as SAM1 and SAM2. And those populations, the blue one is a complement high macrophage population. And you can see here it expresses several complement genes, but it also expresses TREM2, which has been associated to macrophages in many other cancer types, including lung cancer. And then when we looked at the expression of those markers, again, comparing tumor versus non-malignant tissue, you can see they're enriched in the tumor. Then we went back to the mouse model, in this case, a combination of KC, KPC, and IKRAS mice, to see whether we could find similar populations, because the mouse, of course, gives us the opportunity to do eventually functional studies. And just like in human, we were able to define two cell populations in one population characterized by the complement genes as well as TREM2, and the other population in mouse is characterized by KIL3, which actually has no human counterpart, but it also has high expression of plakoglobin 8, which is similar in the human population. Again, just like in human and actually in mouse, we have the possibility, of course, to harvest normal pancreas, which is not just adjacent normal, it's real normal. And you can see that there is an increase in expression of all of those markers in the macrophages in tumors versus normal pancreas. Then we wanted to know whether that population of macrophages also exists in metastatic lesions in human tumors. And since over the past year, we've started sequencing liver metastases of pancreatic cancer patients. Those come from transcutaneous biopsies. And to date, we have fully analyzed data from lesions in 5 different patients. And you can see that just like the primary tumor, we can identify a number of different cell populations, including a lot of infiltrating immune cells. So if we take out the macrophages from the liver and we plot them, we again find that they fall in 2 main populations. And you can see in the Bayesian plots on the right that one of the populations, the blue one, is enriched in those complement genes as well as TREM2. So then we wanted to know, do they actually exist in the tissue? Because that, of course, is just gene expression, and it doesn't tell us if the proteins are expressed and where they are localized. So we did immunostaining, and the cells that are in the insets show macrophages defined by F480. This is mouse tissue in this case, and they're co-expressing C1q, which is one of those complement genes for which there are good antibodies. So then again, one of the things we're really interested in is finding non-invasive ways to know if a patient has a malignancy, and maybe probably the best use of it would be for patients who have been diagnosed and are in treatment, if it could allow us to detect patients who are relapsing. And so for that, we need to know what is happening to the peripheral blood of those patients. So we went back to our sequence data of peripheral white blood cells from pancreatic cancer patients. And this is a feature plot. So it is showing the expression of those 3 genes that define the complement high macrophage population, plotted on, in this case, a specific population of human monocytes. So then we went and compared blood of healthy individuals with blood of pancreatic cancer patients, and you can clearly see that there is an enrichment of all 3 of those markers in patients that have a tumor. This is just showing a broader range of genes that are highly enriched in that population. It also gives you an idea of how the different populations of monocytes in the blood are very clearly defined. They have very specific gene signatures. The arrow points to ApoE, apolipoprotein E, which is another macrophage-derived genes we have recently studied and published together with my colleague Howard Crawford in Cancer Research earlier this year, but I won't go into the detail. I'm going to wrap up. Basically, the conclusion is that we identified a very specific population of complement high macrophages that exist in primary tumor and metastasis. They are recapitulated in the mouse model, and they correspond to a population of monocytes that is enriched in pancreatic cancer patients. And this is sort of a broader summary looking at different immune suppressive signals in pancreatic cancer. We identified TIGIT, which up to that point had not really been on the map as a key driver of immune suppression in pancreatic cancer, and TIGIT would directly inhibit activation of CD8 T cells. But of course, a lot of the immune suppression in pancreatic cancer is driven by myeloid cells. And myeloid cells have different mechanisms of immune suppression, one of which is overexpression of ApoE. Finally, fibroblasts, which is one of my favorite cell types, but I didn't have them in the talk today, but they express a lot of immunosuppressive cytokines. With that, I'll just move on to acknowledging the people. This is a picture in the lab from the time we were in shifts. I have a number of collaborators, including many clinician scientists, as well as basic scientists, of course, funding from NCI and until recently, the American Cancer Society. This is our University of Michigan pancreatic cancer team back from a pre-COVID era picture when we could be together. And this is how we look now. We have a joint lab meeting every Friday, and of course it's now on Zoom. And with that, I would like to probably not take questions or take questions at the end.
Okay, so our next speaker is Dr. Christine Cho from Columbia University Medical Center, where she is the Assistant Professor of Genetic and Development. Dr. Cho received her PhD from University of Toronto, where she worked with Dr. Tech Mack, and then she did her postdoc with Dr. David Chiverson at Cold Spring Harbor. Since joining Columbia, her research has focused on the role of cellular redox regulation and metabolism in pancreatic cancer. She has already received numerous accolades, most recently the Pershing Square Strong Award. And today she'll be talking to us about translation in pancreatic cancer.
All right, good morning, everyone. I would like to start off by thanking the organizers for giving me this opportunity here today to share with you the work that my lab has been doing to try to target the translation machinery in pancreatic cancer and what we've learned so far about mechanisms of therapy resistance. So first of all, it is very well established now that beyond mechanisms of transcription regulation, there are many post-transcriptional mechanisms, including translation, that contributes in large part to gene expression regulation. And this may explain why in certain cell types and contexts, mRNA levels often just explain around 40% of variability in protein levels. And so this has become one area of interest in my laboratory to understand mechanisms of translation regulation. And of course, we know that going from normal to cancer cells, there is an increase in protein synthesis. And this is evident in our organoid panels. Here I'm showing you N representing normal, K representing KRAS mutant pannin, KP representing KRAS P53 double mutant tumor. And in this setting, by metabolic labeling or by sucrose gradient sedimentation to look at polysome fractions, or using Sunset assay to look at in vivo nascent protein synthesis, across the board, it is very clear that protein synthesis is upregulated in the tumor setting. This is, of course, not very surprising. And so the more important or relevant question for us is whether this increase in protein synthesis is transcript specific, or in other words, is there an oncogenic translation program in pancreatic cancer? So to this end, we did deep sequencing of mRNAs associated with translationally active heavy polysomes, comparing a panel of normal ductal organoids to a panel of KRAS P53 double mutant mutation tumor organoids. And what we found upon adjusting to transcription differences, so this is normalized to the total transcriptome, is that there are indeed unique changes in translation. There are some mRNAs that are translationally activated in the tumor setting and some that are translationally suppressed in the tumor setting. And so if there's a program that activates a so-called oncogenic translation program in pancreatic cancer cells, we're interested in determining what governs this program. And we know that translation initiation is the rate limiting step of protein synthesis. And the core, enzymatic core of the complex that regulates translation initiation is the RNA helicase EIF4A. And so we reached out to two colleagues, Jerry Peltier at McGill University and John Porco at Boston University, who's invested a large part of their careers in developing and in studying and designing small molecules that selectively inhibit this RNA helicase EIF4A. And one such molecule that they shared with us is CR131B, which belongs to the family of Wacoglate compounds. And interestingly, what we found was when we applied this compound on tumor organoids to inhibit the translation machinery governed by EIF4A, we saw very robust differences in the translation program upon acute inhibition of EIF4A, which we did not see upon the exact same treatment in normal organoids. And in fact, if we were to compare the translation program in tumor to normal organoids upon EIF4A inhibition, we realized that mRNAs that are translationally activated in the tumor setting now become suppressed upon inhibition of EIF4A. And the opposite is true for translationally suppressed mRNAs in cancer cells. Upon inhibition of EIF4A, now they become translationally activated. So it almost looks like there is a reversal of the translation program upon inhibition of this one single RNA helicase. So what are the enzymes or mRNAs that are regulated by this RNA helicase in this manner? We found that a lot of them are involved in metabolism, and one that we would like to highlight is in the process of glucose uptake. So first of all, we noticed that inhibition of EIF4A potently suppressed glycolysis by metabolic labeling, comparing normal and tumor organoids. This effect is only evident in the tumor setting in KP organoids, but not occurring in normal wild-type ductal organoids. By FDG PET analysis, we found that the inhibition of glycolysis likely occurs at the level of uptake because treatment of mice with this small molecule leads to a very potent and acute decrease in glucose uptake in vivo. So upon examination of the expression of a number of well-characterized glucose transporter in cancer cells, including SLC2A1, 2A3, and 2A4, we found that their expression are insensitive to inhibition of EIF4A, and so we can rule them out in the phenotype that we observed. And surprisingly, we found that a poorly characterized glucose transporter, SLC2A6, actually it's induced in pancreatic tumor organoids, and its expression is dependent on EIF4A. And we confirmed that this glucose transporter SLC2A6 is a translation target of EIF4A by measuring mRNA of SLC2A6 that's associated with translationally light versus heavy polysomes. And importantly, using CRISPR-Cas9 to knock out SLC2A6, we confirmed that it is functionally involved in regulating glycolysis as well as glucose uptake. And so collectively, these data indicate that there is a strong oncogenic translation program that's governed by this RNA helicase EIF4A that contributes to glucose metabolism in pancreatic cancer cells. Now, the next question is whether it is a therapeutic opportunity. In comparing the IC50 of a panel of normal to tumor organoids to this RNA helicase inhibitor against EIF4A, we found that the tumor organoids are at least 1 log more sensitive to the small molecule inhibitor in vitro. And this also translates in vivo. In a short-term tumor kinetic studies in KPC mice, we can see that inhibition of this translation program using CR131B does significantly suppress the pancreatic tumor growth in vivo. And we confirmed that the drug actually did enter the tumor by doing an in vivo sunset assay to measure nascent protein synthesis. Inhibition of this translation program using this RNA helicase inhibitor does not induce systemic toxicity, and this is through measurement of serum levels of ALT, AST, as well as body weight. There's no significant difference between the two groups. And so this prompted us to take this study into a longitudinal setting to examine whether EIF4A inhibition is a therapeutic opportunity or not. And the result was quite disappointing because even though the single agent small molecule was able to prolong survival of KPC mice, the improvement was extremely modest and very limited, which led us to question whether there was development of a resistance or adaptive mechanism. And so to answer this question, we generated a pair of two pairs of pancreatic tumor organoids that are either sensitive or resistant to EIF4A inhibition. And we found that while these organoids develop resistant to EIF4A inhibition, they remain sensitive to chemotherapeutic agents, meaning that this resistance mechanism is highly selective for inhibition of this RNA helicase. We confirmed by Sanger sequencing that the resistance line did not develop a genetic mutation in the drug binding pocket. So this means that there's alternative reasons that explains the resistance mechanism. And given that the translation program governed by EIF4A is involved in regulating metabolism, this prompted us to perform a metabolic drug screen using 240 compounds targeting metabolic enzymes to identify mechanisms of resistance. And to our greatest surprise, we found that there were 4 top hits in this screen, including ML210, ML162, RSL3, and Erastin. All of them are inducers of ferroptosis. And what this drug screen tells us is that induction of ferroptosis render the resistant cells more sensitive to EIF4A inhibition. In other words, treatment with these ferroptosis-inducing agents would resensitize resistant cells to our inhibitor against EIF4A, as shown here. And so there are 3 hallmarks of ferroptosis, which includes defective lipid peroxide repair, redox active iron, as well as PUFA phospholipids. And for the interest of time, I will just tell you that our experiments allow us to exclude these 2 properties or hallmarks of ferroptosis. And so we focused on looking at changes in the lipidome. We teamed up with Brent Stockwell here at Columbia, and we performed lipidomics to compare our sensitive versus resistant pancreatic tumor organoid lines. And shown here are the principal component analysis showing clear separation of the 2 groups. And when we look at the identity of the lipids that undergo or exhibit differences in our sensitive versus resistant lines, we see that they enrich for one specific class of lipid species. These are ether-link phospholipids. And so this is the, and in addition, so these are results from organoids, and we want to make sure that whatever is happening in the organoids, it's also reflective of what is happening in vivo. And so to this end, we have been developing mass spec imaging approaches using either MultiTOF or DESI that would allow us to examine lipid species changes specifically in tissues. And so we're now in the process of characterizing the lipidome changes in tissues, as well as to functionally characterize why ether-link phospholipids contribute to resistance to EIF4A inhibition. And moving forward, what we would like to do is to characterize ether-link phospholipids in pancreatic tumor metabolism, as well as also to ask whether inhibition of enzymes governing this lipid metabolic pathway could synergize with inhibition of EIF4A. We're also working in characterizing if there are translation programs that's activated beyond just normal to tumor cells, but going from tumor to metastatic pancreatic cancer cells. And so with this, I would like to close and acknowledge all the hardworking people in my lab who made this possible, and most importantly, all of our collaborators who made this work possible. Thank you very much for your attention, and I will be happy to take questions.
Thank you, Christine, for staying within your time frame. But please stay around until the end of the session if we have any questions from the audience. So our next speaker is Dr. Colin Weeks from the Mass General Hospital, where he's the Director of Medical Oncology Research for Pancreatic Cancer and the Director of Cancer Center Grand Round Program. He's also an Associate Professor of Medicine at Harvard Medical School. Dr. Weeks received combined MD PhD from University of Nebraska Medical Center. He did his residency at University of Alabama, Birmingham, Medical Oncology Fellowship at Hopkins. And prior to joining Harvard, he was a faculty at the University of Colorado. Dr. Weeks has a long history of working on translational research involving novel therapeutic for pancreatic cancer. Today, he'll be talking to us about drug resistance in pancreatic cancer.
I thank the organizers for the opportunity to speak with you all today. So I was asked to talk about mechanisms of resistance to chemotherapy, and I'm going to talk about some work that we're doing on targeting tumor cell plasticity in that context. These are my disclosures. So I always start with this slide because I think it's very humbling, as has been pointed out previously. There's multiple cell types that are involved in this disease and multiple pathways that we have to think about as we think about response to chemotherapy. I think this slide is also very humbling. So this is the slide that shows the 2 active regimens for pancreas cancer that were published in 2011. So it's been a decade since FOLFIRINOX and gemcitabine and Abraxane were approved for the treatment of pancreas cancer, and we still have not been able to move beyond that. And so I've always thought about, do we understand the consequences associated with chemotherapy, and can we use those consequences to then target resistance? So during the same decade that I just referred to, there's been a lot of work, as you all are familiar with, in terms of molecularly characterizing these tumors. And basically, there are sort of really 2 cell types that we think about, which is the classic or epithelial cell type, or the basal and quasi-mesenchymal cell types. And this has now become clinically relevant based upon the results of the COMPASS trial. And so in this trial, patients with metastatic or locally advanced pancreas cancer were treated with FOLFIRINOX, and then they retrospectively asked if they are GATA6 RNA in situ hybridization positive, i.e. epithelial, do they respond to FOLFIRINOX? And what they showed was that those patients who were epithelial, seen here in purple, preferentially respond to FOLFIRINOX in panel A, and then that was associated with an improved progression-free survival, thus suggesting that those patients with the epithelial phenotype are the ones that should be treated with FOLFIRINOX. And so simultaneously, work at our center at MGH, Dave Ting, demonstrated that in patients with locally advanced disease that 2/3 of those patients have epithelial phenotype at baseline, and then that converted to 2/3 of the patients having mesenchymal phenotype at the time in which we go to surgery, and these are patients who are receiving neoadjuvant therapy. And he's developed this RNA in situ hybridization 2-color model in which he can determine the percentage of cells that are epithelial versus those that are mesenchymal. So it gives us an opportunity to sort of quantify the relative percentage of those phenotypes within a given tumor. He went on to show that in patients, as you treat with FOLFIRINOX, that what you see is this transition from an epithelial phenotype, which is demonstrated in red, to more of a mesenchymal phenotype, which is demonstrated in the blue. And conversely, if you treat cells with gemcitabine and Abraxane, the reverse is true. So if you'll go from a mesenchymal to an epithelial phenotype. And that's just depicted here. And so once again, when we treat with FOLFIRINOX, we're transitioning to a quasi-mesenchymal phenotype, and that may be associated with resistance. And then conversely, in those patients who have a quasi-mesenchymal phenotype, as we expose them to Abraxane, we transition to an epithelial phenotype. He went on to show that, in fact, the cancer-associated fibroblasts do dictate this to some degree. And so what he did is a mixing experiment where he took an epithelial cell line and co-cultured that with cancer-associated fibroblasts, and showed that with the cancer-associated, when you use different ratios of cancer-associated fibroblasts, you increase the growth parameters of the tumor. And then in addition to that, if you look at those that had more fibroblasts, the 30 to 70, there's an increase in the metastatic potential of those cells. He then went on to demonstrate that, in fact, TGF-beta is an important component of this. And so what he showed was that when he was able to co-culture these tumor cells with cancer-associated fibroblasts, that you get an increase in TGF-beta. If you block that TGF-beta, then you decrease the growth of what he's calling these dual positive cells. And these are the cells that have mesenchymal phenotype and have the ability to proliferate. And in fact, if you do the reverse, which is you expose the cells to TGF-beta, you can then induce this process as well. And so then this gives us a paradigm in which, in fact, if we treat cells with FOLFIRINOX, they'll transition to epithelial, and that's TGF-beta dependent, and that's potentially cancer-associated fibroblasts mediated. And then if we inhibit TGF-beta, then presumably we'll keep the cells in this epithelial phenotype and therefore potentially keep them responsive to FOLFIRINOX. And so I'd like to just show you how we're going to clinically try to prove that. And so prior to me coming to MGH, there was a lot of work done by Rakesh Jain's lab and the team that was there looking at repurposing losartan or angiotensin receptor blockers for treatment for pancreas cancer with the idea that this inhibits the production of TGF-beta. And so work there had shown that, in fact, when you use losartan as a chemotherapy agent, that about 2/3 of the patients are able to undergo resection in patients who have locally advanced disease. In associated with that, we also see there's a change in the immune infiltrate associated with that, where now you have increased CD8 positive cells. And we've now just completed, I think last week, hopefully, a randomized study in which patients were randomized to receive FOLFIRINOX, losartan, and losartan plus a PD-1 inhibitor. But losartan does many different things. And so here we're going to use a specific TGF-beta inhibitor, and we're working with Novartis to develop a study in which we're going to use their specific TGF-beta inhibitor in patients with locally advanced disease. So the study is designed in which patients would receive FOLFIRINOX as neoadjuvant therapy followed by radiation and surgery versus the addition of the TGF-beta inhibitor followed by the addition of TGF-beta inhibitor with immune checkpoint blockade. And importantly, we're going to be doing the RNA in situ hybridization assay both at baseline and then as we sample patients throughout their treatment. So we'll be able to assess what happens to this transition with therapy and is TGF-beta altering that process. In addition, we're going to be using peroxypyl MRI imaging to look at macrophage trafficking while the patients are being treated on therapy. And then as we're sampling these patients, we can then do also multiplex immunofluorescence to understand what is the macrophage polarization associated with that, what are the immune cells that are being transiting into the tumor, and what are their association. In another set of experiments, Holly Bruton's group demonstrated that GSK3 beta also is important for this transition to mesenchymal phenotype, so blocks EMT, and that's associated with glycolysis. And so we've designed another study to address this question. And this is now using losartan once again as a TGF-beta inhibitor, but in this case, adding GSK3 beta inhibition as well. And this is in patients with metastatic pancreas cancer. Now we've designed this study to for patients to be treated on what I'm calling complete therapy, which is FOLFIRINOX plus whatever biologic agent, then transitioning to maintenance therapy, and then going back onto complete therapy. And the hypothesis here is that in patients who are receiving FOLFIRINOX, if in fact we're maintaining this epithelial phenotype, we should see an improved progression-free survival. So that's the primary endpoint of this study. We also know that maintenance therapy is effective in pancreas cancer patients. And so can we, after we've debulked the tumors, can we then keep them in sort of this pressured situation where we're sort of altering the tumor microenvironment to then support suppression of these tumor cells. And then when these tumors grow out, it's probably not a result of resistance to therapy, but maybe it's more of a fact that the tumor just overcame the pressure that we put in place. And so then can we debulk these tumors again by putting them back on the complete therapy. And once again, we'll be using the RNA in situ hybridization technique to look at what's happening to the transition between these different molecular phenotypes as we're treating the patients. Also looking at peroxyl MRI to look at macrophage infiltration as well as macrophage polarization. And then also in addition to that, doing digital spatial analysis as well. And so I'd just like to sort of make this last point, which is, a lot of times in pancreas cancer, we do these clinical trials and the drug doesn't work. And so that drug is then dead. But what we really need to do is really think about doing clinical trials and further investigating the tumors as we're treating these patients so that we have an informed understanding of why the drugs didn't work or why they did work. And ultimately be able to do these sort of second generation trials that will allow us to hopefully be able to move the needle forward in this disease. So that I'd like to thank you. I'd like to thank my large collaborative team at MGH, as well as members of our friends at Hopkins, Lei Zhang, and then Holly Bruton in the UK. Thank you.
Okay, we'll open up this session for some questions. I'd like to keep it brief because I know we're running behind, but please come up. Do we have the other two speakers on live?
Yes, I'm here. Good morning.
Is there another microphone?
Yeah, go ahead.
Oh, okay. So I've seen a couple of speakers now talk about losartan, and there's, and I wasn't aware of its ability to inhibit TGF-beta. I'm aware of a different basic mechanism that looks great in mice and lung cancer. But so my question is, are you aware of any human studies in which losartan has shown some benefit?
So, so we have a published study that's in JAMA, and that showed that in patients who have locally advanced pancreas cancer, if we treat them with losartan plus FOLFIRINOX in the neoadjuvant setting, that we're able to convert about 2/3 of those patients to resectable disease. And now in long-term follow-up, we show that in those patients who are resected, those patients have a long-term disease benefit. So the median survival of those patients is around 40 months.
And with FOLFIRINOX, it's what, in the 30s?
Right,
Yeah.
and also most of those patients are not resected, right? So the true benefit associated with sort of our therapies for localized disease for pancreas cancer is resection. And so if in the patient population with locally advanced disease, those are unresectable patients. And so we're converting those patients to resection. Now, we just finished a randomized trial that I don't know the data yet. It's just, I think we enrolled the last patient last week. So hopefully we'll have some additional information in the next year or so with regards to the impact of losartan in this context.
Okay, thank you.
I have a question for Christine Shaw, the online presenter. So, under translation initiation complex components changes during hypoxic conditions, I wonder your 4A inhibitor is able to inhibit tumor organoids grown in hypoxic conditions.
That's a really good question. So we haven't done the experiment where we culture organoids in hypoxia and treat with the drug. But one thing I can tell you is that the oxygen tension in Matrigel is not the same as atmospheric oxygen tension, because when we compare activation of HIF1, for example, in pancreatic cancer cells that are grown in Matrigel versus in monolayer, there's a much higher level of HIF1 activation. And so our suspicion is that within the Matrigel dome, it already is under a lower oxygen tension. And so whenever we try to put these organoid cultures into 3% oxygen, what we tend to see is that they experience anoxia and a lot of cell death happens. So we couldn't really do that experiment you described.
Thank you. One quick clarification with Dr. Madeleine. There's a complement C1q rich macrophage. I thought macrophages have complement receptors. Do you think this is the receptor bound C1q, or you?
So those are the genes that are expressed in the macrophages. So then whether the protein we see came, we can tell where the protein came, but they have it in vitro too. So they have the transcript, they make the protein.
Agnes from University of Miami. My question to Marina. So, it's so important in cell-to-cell communication and the ligand to receptor interaction studies, and saw your slide. So my question is, this is the thought process that if the ligand and receptor interaction, how different based on the mutation status and any signaling pathways variation?
So you can map those differences in signaling pathways, and there's definitely upregulation of a number of signaling pathways, which have already been described in pancreatic cancer. The other thing for those studies is that the way we chose to go about it was looking at, we call it a differential interaction if the ligand is changed, because receptors don't usually change the same way. They actually often are downregulated when the pathway is activated. So this is how the data is processed, but of course you can process it in many different ways. So that's both the beauty and challenge of bioinformatics. You have to, decide how you're going to analyze data and ask questions.
Thank you.
Josh Tata from the University of Miami. I have one question, Dr. Pascal Di Magliano, one for Dr. Weeks. For you, Dr. Marina, so complement on macrophages is an interesting concept. So is there opsonization or phagocytosis going on in the tumor microenvironment? And secondly, do you think that it's also a surrogate for the activation status of the macrophage, either classical or alternate? And for Dr. Weeks, how does one explain the fact that some patients with FOLFIRINOX, especially in the modern era of doing more and more neoadjuvant therapy, have complete pathologic responses? Does this reductionist approach of biomarker-driven chemotherapy selection just speak to intertumor heterogeneity and the fact that we're not just capturing it?
So, okay, the short answer is we don't know yet. We are, all of it is possible. So those macrophages are a subset of tumor-associated macrophages, and they're not, they're not the only tumor-associated macrophages. So they're not the only M2-like macrophages. So it's a subgroup. It could be that reflects activation. We don't know. We are doing functional experiments right now, but we don't have the data yet.
Yeah, so with regards to the tumor heterogeneity, I think at the end of the day, those patients who respond, I think, TGF-beta is probably one component of that, right? There's other components of that regulate EMT. And so to what degree the different components are involved in this process. And there's other components, there's the immune system, there's other things of that nature. So I think it's multifactorial. I don't think it's just there's one answer, but you're clear there are patients who do respond very nicely to FOLFIRINOX, but that's, I would say, is not the norm.
This will be the last question.
Hi, I'm Anshu. I'm from UNMC. So my first question is to Dr. Medina. That is very technical question. That have you done kinetics experiment in the co-culture of organoid with the T cell to see checkpoint molecules overexpressing after some time, not in the short times like 24 hours?
I'm not sure I heard the question. Could it be repeated, please?
So my question is, have you done kinetic experiments where you incubated or co-cultured organoids with the T cell to check the overexpression of checkpoint molecules, like coming after a time, not immediately?
Yes, thank you. Good question. We have not done kinetic studies in the sense of collecting the cells at different time points, but in the final cells, when we collect it after expansion, which is about 2 weeks, what we see is that, yes, there is checkpoint expression, but it's very different from one patient to another. There is a lot of heterogeneity, which indicates that probably the idea that PD-1 CTLA-4 is going to be universally effective is not is not real. We see TIGIT, we see LAG3, we see other things. So there is room for combining these cells with checkpoint inhibitors. I think that will be more effective if it is to some extent individualized.
Thank you. This last question to Dr. Vasco. So you showed TIGIT expression like as a suppression molecule. Have you seen any T cells which are overexpressing this, like co-expression with other checkpoint molecules on those T cells?
Yes, that's not the only checkpoint. There is a wide variety, and we actually also look patient by patient and cell by cell, and it's very heterogeneous. So, but TIGIT was the one that correlated the best with exhaustion markers, but those cells also express other immune checkpoints.
Thank you.
All right, unfortunately, due to the time, so we just have to close the session. We want to thank all the speakers for their wonderful presentations and the audience for your participation.
We have a lunch downstairs in Eden Rock 1, and then please have your lunch quickly and then go visit the poster sessions and watch the posters. At 2 o'clock, we will meet in this room for business meeting, and at 2:30 scientific session will start. Thank you.
Yeah, no, I think